Pleasure to be here and speak. I'm glad to see so many people excited about the brain. As a neurologist, I see people with brain and nervous system complaints. So they come in with a certain problem, and our job is to use our history to go through the problem and decide, is this something that's a certain cause or another cause? And then we use our examination to decide where it is in the nervous system. So the analogy for that, you'll see I like analogies, is a flickering light bulb. So if you have a flickering light bulb in your house, I tell patients, that could be your light bulb, it could be the wire to the light bulb, it could be the fuse box, or it could be the power plant, and that's basically neurology. Sometimes those things are all fine and you've got a power surge going on. In neurology, what we do is we try and figure out where the problem is in the circuit. So I'm going to use a few more analogies here to explain to you how I think about degenerative brain disorders. So these are things that gradually, progressively get worse over time. I'll use as an example Parkinson's disease. And I want you to remember three things here. I'm going to talk about the tree, the garbage bin, and the barrel. So we'll come back to each of these in turn. The tree is an analogy for how the brain normally changes its function over time. The garbage bin is how the brain responds to wear and tear changes over the lifespan. And the barrel is what happens when there's a degenerative brain disorder going on, the rusty barrel. So the tree, the bin, and the barrel. We'll start with the tree. So here's a resilient little sprout coming out of a place where it should not be growing. So this is an analogy to remind you that brain health, or the health of a plant, is partly to do with the plant itself, which is sort of like genetic factors, and it's partly to do with the soil or the conditions. And you can think of your brain health in the same way. There are some things you can't change about the genetics of how your brain is built up, but you can give it the conditions to thrive. And in some cases, those are things you can't modify or change, but some things you can. Although you can't change the kind of plant you are, sometimes you can give a plant better conditions and it will do pretty well. So this is a nice little figure here. You'll see the original creator link down there. And this is one way you can think about brain function over time. So unfortunately, age is still not something we can fix. If you think about trees, in this analogy here, the density of the leaves is the health of the tree. Over time, seasonally for trees, but over time for people, the density reduces. So in the same way, your brain is only designed to last usually, say, 20 years, then you get eaten by a bear or something like that, right? But now we're living into our 60s, 70s, 80s, 90s, sometimes even longer, and there are wear and tear changes associated with that. Unpacking that a little bit, so if you think about how to strengthen and grow a tree, you've got to give it those right conditions. And similarly, if you want to optimize your brain function over time, you can do a few specific things that help with that. So the level of education that you have has been shown to help with the long-term function of your brain and your brain health. Different challenges and experiences that you expose your brain to will allow you to create new connections and new strategies for signaling in the brain. And activity and nutrition are very important. In fact, exercise, we'll come back to, is probably one of the most important things for your long-term brain health. But over time in adulthood, after about the age of 20 or 30, you gradually start losing neuronal density. So your neurons, those signaling cells within the brain, they do slowly wither away. Every couple of days, you're losing probably 10 or 15,000 neurons. Whether or not you have head injuries, or you're a football player or a boxer, or you have strokes. We look at that as the natural age-related change. And on top of that, sometimes there's a second thing, like a disorder or disease, that accelerates that. So sometimes we can fix those things. Here's a couple of pictures, MRI pictures from two brains. So on the left-hand side of the screen, you see an example of a younger brain. So this is a brain of someone around the age of 30. The top picture on the left is called an axial picture on an MRI, or a transverse. You can remember, if someone has an axe, they cut a tree like this, and that's the way the slice is taken. So it's kind of like this. In this case, it's through the white matter and the hemispheres up near the top. And then you can see on the right-hand side of the screen, the same kind of axial picture is done there in an older patient. And in this case, using my arrow here, you can just see the little darker spaces are fluid-filled spaces. The gray spaces are areas where there's brain tissue. And there's more fluid-filled spaces and less brain tissue. And that's what we call atrophy, or age-related change. In some cases, that can be a disorder. In other cases, it's part of an age-related thing. And so there's a spectrum of that change over time. And as a neurologist, if someone comes with a memory problem to the office, we try and look at this sort of thing. Is this something that's age-related, or is it beyond the pale? Is there a disorder or disease underlying this? The lower images here are just a different way of looking at the same thing. So axial is like this, and this is what we call a coronal image. If you think of sort of an old-time crown looking like this, that's what coronal means. And so these pictures are sort of right up and down like this through the years, basically. Here you see, again, the younger brain, it looks a little more full and dense in there, and then there's a little bit more space in the older one. So that's the concept of atrophy. That's less leaves over time. Looking again at the tree, we're going to transition from the tree analogy into a little bit more about what happens inside neurons. And yes, I am going to show you a picture of a neuron cartoon. I can't help myself. Neurons they're basically just information exchangers. These are like wires that are sending signals between each other, and it's not just one wire to the next. It's a very complicated web of wires. I've taken off most of the labels here, and I've added some extra cartoons. So you can see here, this little wire here is called the axon. So this is basically the signaling pathway that a message goes down from a neuron to communicate with another one. And you can see there's little cars here. I've shown these little cars as things that are delivering a message. So you create systems and packages here in the cell body, and you're going to deliver them down to release them to send a message to another cell at the end terminal there. And if we zoom in a little bit here, so this is right at the end of one of those terminals now. So those cars are dropping off a bunch of little boxes there, or packages, and the packages have letters and other documents and things inside them. As I told you, I like analogies. But basically, we're sending messages from one cell to the next. In order to do that effectively, you've got to have a good highway here for the cars to travel down, and you've got to create the messages well, and you've got to have good little packages. And so I'm telling you this because it's sort of relevant to degenerative brain disease. If we look inside the inner workings of the neuron cell, I've drawn some little factories here. So these little factories are producing the stuff. They're producing the messages that you're going to send to the next neuron. And you can see they're creating smoke here. There's some smoke coming out of these smokestacks. That's because it takes energy to do these things, and sometimes there's waste products generated as a result. Either if you generate too much waste in the cell, or if the cell is not good at handling waste, then you're going to wind up with a bit of a garbage problem. And we think that for most degenerative brain disorders, this kind of a garbage problem is pretty important. Now we're going to move on to the garbage bin. I moved to Kelowna about 18 months ago now, and when I got here, I got the garbage bin. I've got 3 kids, so we produce a fair amount of waste. Unfortunately, we're trying to reduce it, but we found we don't have enough space. So the city of Kelowna says, well, if you want a bigger bin, you can pay for the bigger bin. We give you more capacity, and then you just keep paying for the bin. You can fill it up a little bit more. If you think about the waste analogy in your cell, you can't buy a bigger bin. You sort of got what you're born with in terms of your capacity for waste removal. The idea here is if you fill up your waste basket in the cell, then you can run into trouble. And the garbage bin will come along, but it won't clear everything out. Are you with me? Are we still talking about brains? I hope so. Okay. All right. So here's an example. So Parkinson's disease. What is Parkinson's anyway? It's classically thought of as a movement disorder because when people come in with symptoms of Parkinson's, they're typically what we call motor symptoms. So they come in with slowness of movement or tremor or stiffness or problems with their walking or falls. And so in medical school, medical students learn that as TRAP. So tremor, rigidity, akinesia, which is another word for slowness, or postural instability, or difficulty controlling posture and balance. So TRAP. And then this little cartoon here illustrates that for 2 patients with Parkinson's, the emphasis may be really quite different. So some patients may have mostly tremor. They come in with a lot of shaking, and they say, well, what is this shaking from? And we go through and we say, well, it may be Parkinson's and so on. But other patients may come with no tremor whatsoever. And they may say, well, I don't have any tremor. How could it be Parkinson's? Well, in fact, many patients don't. It's actually not just one disorder, but it commonly presents with a little cluster of features that we look for. And then the diagnosis is based on our clinical assessment, so the history and the exam, looking at the light bulb problem. Some of the other things people complain of, the softness of their voice, their face may be less expressive or less animated. They may have trouble with hand tasks, like 2-hand tasks, cutting food, doing up buttons or clothing, handwriting, turning in bed, getting up out of a car seat that's quite low to the ground or a deep sofa, something like that. Those are the motor symptoms. There's been increasing attention in the last couple of decades, particularly the last 10 years or so, about other symptoms that are also features of this illness. So it's not just something that creates these motor symptoms. It also creates sleep problems, mood problems, cognitive or memory problems, problems with psychosis, which could be hallucinations, for instance, and then plumbing problems, so bladder problems, bowel problems, blood pressure problems. So it's actually very complex. And like the emphasis can be a bit different with the motor symptoms, some patients may have these things in spades and others not at all. So we have to look at all of these things as part of the overall picture when we assess these patients. What I'm going to tell you here is something that's rather new in terms of the conception of Parkinson's disease. It's classically just described as a disorder of dopamine in the brain. But what I'm going to suggest is that dopamine, one of those neurotransmitter chemicals, is responsible for a lot of the motor symptoms. When it's not working right, you get stiffness, slowness, tremor, etc. But that's not the disease itself. It's a symptom of the disease. So the disease is actually a problem with the metabolic function of the nerve cell, the waste disposal system. And when the waste disposal system is overwhelmed, eventually those dopamine cells are unhappy, they don't work well anymore, and your brain can compensate for a while, but eventually you run out of room in compensation and you exhibit the symptoms. I'm going to explain that a bit more, and you may say, what is this picture you've got up there? Well, it's again a coronal picture, so it's another one of those coronal slices through about the same area I showed you those other scans. And here, I'm showing you some key brain structures for Parkinson's. These things here, these deep areas that are blue in this cartoon, are called the basal ganglia. You don't necessarily have to remember that, but the basal ganglia are like a computer to control or coordinate movements. Those are things that help you decide when to initiate a movement and how to select among different choices of movements. I risk getting a little bit off topic if I tell you it can do the same thing to some extent for your thoughts and other things, your emotions. So if you have a choice of different thought processes, the basal ganglia can help you select among them or tell you when a thought is appropriate or not. These little wires down here represent the dopamine cells, which live a little bit lower down in the back of the brain, in the brainstem. And they're like fine little microcatheters or little tubes that just spit out a little bit of dopamine right where you need it, right when you need it. It's very exquisite. Unfortunately, we don't have great medications that can replace that specificity and that exquisite control. Because if we give you a medication, it doesn't just go right where it's needed or right where you're missing it. It goes all over the place, not just in the brain, but your whole body. So this is one of the big problems with medications taken by mouth for a very focused problem. These are similar slides to the cartoon here, looking at this part of the brainstem called the midbrain. And you see where these little wires are coming from is shaded a bit darker here. That's an area called the substantia nigra. And in the brain at autopsy, a normal healthy one looks like this. That's why it's called the substantia nigra, because it's darker. It's got pigment in it. And those pigmented areas are dopamine cells. In a patient with Parkinson's disease, you can see, where's the pigment? So this is how pathologists figured out what's going on in these brains many years ago when we first identified these problems. It's something that's visual, and when you dig into it a little bit deeper, you find out, well, those are dopamine cells and they're missing. So it turns out the problem in the midbrain, there where the dopamine cells live, is accompanied by accumulation of these clusters of proteins that are called Lewy bodies. This is extra waste that your brain cells have not been able to get rid of. And when the brain is overwhelmed, its waste disposal capacity, those little clumps of protein stick together, and after a while, the cell gets unhappy with that, and it doesn't work well, and it dies. So we see these Lewy bodies in the midbrain in patients who have the motor symptoms of Parkinson's. And when patients have other problems with memory function and things that develop some of those non-motor symptoms, they may relate to Lewy bodies that are elsewhere in the brain. It was very interesting to learn in the last decade or 15 years that Lewy bodies seem to have a spread through the brain in these disorders. So it seems like they start somewhere, and then over time we see them kind of expand and spread through the brain. That spread is also seen in a number of other degenerative disorders, which I'll come back to. Please don't think you need to learn about this. I use this as a placeholder to show you how complicated things are and to emphasize that some of the discoveries in the science behind Parkinson's are specifically about that waste disposal system. So these little numbers up here are, this alphabet soup is basically a list of different genetic causes of Parkinson's that have been identified. And it turns out that all of them in some way have to do with the packaging, transport, or degradation or disposal of things in the neuron. They have to do with basically the garbage disposal system or the garbage truck or the highway along which it travels. So when you have a problem with the protein itself, that's one thing, but if all this system is working perfectly, you may be all right. But if the highway is not quite in great shape and the car has a busted wheel and you've got extra waste sitting around, then you're going to run into problems with too much waste. Still with me? Are we still talking about the brain? Okay, good. All right, so just to show you, the reason I discuss this in terms of Parkinson's is just out of personal interest, but there's remarkable analogy for a lot of the other degenerative disorders in neurology. So in Alzheimer's disease, there are different proteins that spread. The green brain up here shows the spread of a protein called tau in Alzheimer's disease. And then there's another thing called beta amyloid, which has another pattern of spread through the brain as the progression goes along with Alzheimer's disease. You may have heard of ALS, the ice bucket challenge thing. So this is also called Lou Gehrig's disease. It's also a progressive degenerative disorder, except it targets a different system. It's the motor neurons. And it's the motor neurons in the brain and the spinal cord and the lower motor nerves. It turns out that there's a spread of abnormal protein accumulation in ALS as well. So there's a lot of things that are very similar between these disorders. They have a lot of similarities in terms of the time course. If you go back to the light bulb, this is a problem where your light bulb's not flickering. It's just not as bright as it was last week. And it doesn't matter if you change the light bulb, the other light bulb's not as bright anymore. And over time, it gets dimmer and dimmer. If it's flickering, but it's still just as bright all the time, well, that can't be a degenerative disorder because it's still just as bright. But if over time, the light bulb is having more and more problems, that indicates there's a progressive problem. So that's kind of the process we go through in neurology in sorting these things out. Okay, so that's the bin. So we've talked about the tree, we've talked about the bin, and now we've got the rusty barrel. So this is another analogy, if you're still with me and not nauseated by them by now, or by the movement. I like this analogy because a rusty barrel is something that's been going on a little while when you notice there's a problem. So you notice there's a problem with your barrel when it starts leaking. So there's stuff coming out, hopefully not good wine. But if there's a hole in the barrel from rust, it's been going on a long time. And we think neurodegenerative disorders similarly are very slowly progressive. They require the right conditions, so it has to be made of metal, it has to be exposed to water and oxygen in order to rust. So similarly, the brain has to have the right genetic problems with waste disposal, and in some ways it has to be exposed to the right stresses through your life. A combination of those things creates the problem. Like the rusty barrel, you may not know that this is going on until you spring a leak and you start having problems with your walking or a tremor, but perhaps it was actually going on for many years at that point. If I'm to kind of summarize that in more plain language, the way I think about these degenerative disorders is you have some genetic risk for them that's predetermined. We don't know everything there is to know about this either. We know for Parkinson's there's dozens of genes that increase your risk, and some of them may be a little bit, some of them may be a lot, and some people have those genes and they don't get the disease. Along with that, there are risk factors and protective factors that kind of adjust how likely or unlikely you are to eventually develop that diagnosis. There's environmental stressors, things that are around you in the world that you have no control over, as opposed to things you do. And some combination of those may result at some point in too much waste, the cells stop working well, and if that progresses for a while, they start dying off. For a while, your brain is very good at filling in the gaps and compensating for this, but after a while, the compensation is overwhelmed by cell loss, and you start getting symptoms. So that's how I think about these disorders. And the problem is, what we do in terms of medical treatments for these, for the most part, is we put our finger on the hole. We treat the symptoms, but not the rusty problem. So in BC, Parkinson's disease is more and more common as people age. You can see here women are in red and men are in blue. So there's more Parkinson's if you're a man. We think that's because estrogen is protective in some way. But as you can see, the curves go up as age goes up. We don't have a fix for age, but that's probably the most important risk factor. If we were all able to live to 150 years, I think we'd all overwhelm our systems in some way. And so it's not necessarily a fault in your genes if you develop one of these things. It's a combination of things, and it's complicated. What do we know about these risk factors and protective factors? Well, this is fascinating. There's more and more about this all the time, but we know higher risk with pesticide exposure, which is an issue I think particularly germane to BC and the interior. Head injuries are not good. People recognize this, of course, because of Muhammad Ali, one of the more famous patients with Parkinson's disease. Rural living, well water drinking. These are also things that increase risk of Parkinson's if you look at big populations of thousands or millions of people. What reduces your risk? Nicotine. Don't start smoking, please. There's nicotine in some foods as well, you may be interested to know. But the drawbacks of smoking far outweigh this benefit. Coffee is good, though. I have no problems with that, although it may make tremor worse. Some anti-inflammatories and other medications are also good for protection, and exercise is a very potent protective factor. This is a map in the US that shows prevalence, and there are some conclusions you can kind of make based on this, but there seems to be some areas where there are clusterings of higher incidence of Parkinson's disease. The reasons for that are speculative, but interesting. We can talk about that more after in questions if you wish. What can you do to prevent this from happening? Well, there's no miracle grow for the brain, but you have to give your tree the best conditions. So you have to exercise your brain, and you do that by challenging it, by doing new things. Learning new things is particularly good for your brain. And activity, nutrition, exercise, physical exercise, probably one of the most potent things to increase brain growth factors and density of connections. So more leaves. I would say it's very important to promote brain vascular health. What do I mean by that? Well, stroke is extremely common, and covert strokes are also extremely common. So these are areas of damage in the brain that you may have not had any symptoms from. And they may accumulate over time if you're a smoker, high blood pressure, high cholesterol, diabetes, etc. And so if you can protect your brain from that, you'll prevent yourself from getting holes shot in the barrel. If you have a slowly progressive problem, and on top of that you start having stroke damage, it really makes the problem a lot worse. In general, I'd say what's good for the heart is good for the brain. That includes exercise. And don't overfill a bin, so we're learning more about this, but probably things like head injury are good to avoid. So what do we do here at the big hospital? So this is a little cartoon of KGH. It's not quite on with the accuracy, but you get the idea. So we have a movement disorders program now, the Okanagan Movement Disorders Program, and we're gradually growing this. Mostly what we see is Parkinson's disease, some other movement disorders, and it's been supported by Interior Health, by UBC Okanagan, by the Parkinson's Society of BC, and the local support group. It's been a big group effort, and it's still very much a work in progress. We are going to undertake some new research, so we're going to start doing a program of activity recording and symptom monitoring, which I'm going to call ParkinSmart. We should be starting a clinical trial as soon as hopefully next week or next month, which is fantastic with the help of Okanagan Clinical Trials here. And there are some exciting opportunities for research collaborations at UBC Okanagan. I think we'll leave it at that for now. Thanks.
So while Bud gets us set up, let me reintroduce myself. I'm Phil Barker from UBC. And as Bud said, I'm actually from the region. I grew up here, graduated from KSS about 4 years ago. And it's really great to be back here in Kelowna. I was in Montreal for 20 years. And what's going on in the Okanagan Valley at UBC is incredibly exciting. And it's great to be back and be part of that. Having said that, I realize I've been brought here under false pretenses because I thought this was a course called MiniMed, and now I realize that you're actually evaluating me instead of me evaluating you. So, not too happy about that, but I'm going to carry on. Anyways, seriously, it looks like it's great to see so many people here. My job is to tell you a bit about Alzheimer's. I don't come from the clinic. I'm not an MD. I come from the basic science world, sort of in the trenches, trying to figure out what's going on in neurodegenerative disease. And over the last few years, the focus of my lab has really been on Alzheimer's in particular. There's a few lessons that I want to communicate to you tonight. The first one is that your brain is constantly changing. And if Daryl and I have done a good job tonight, the brain you leave with today will be a little bit different than when you walked in the room. It'll change by tomorrow, and it'll be different a week from now. So I want to talk about why that is and how that works in less than 20 minutes. Then I want to talk a little bit about how that relates to what goes wrong in Alzheimer's disease, and really the current thinking from sort of the front edge of the field on what we think is going on. And then give some indication of where we are with regard to treatments, and then talk a little bit about what, if anything, we can do to slow the progression of this devastating disease. I told my son Liam, who's 7 years old, that I was giving this talk tonight, and he said, "Dad, you have to lead with a joke." So I'm going to do that now. So it's actually a riddle. What do you call a cow with no legs? Ground beef. What do you call a cow with no legs? Ground beef, okay? So thank you, Liam. All right. So just reiterating a little bit of what Daryl was saying, when you're a neuroscientist and you look at the brain is full of a whole bunch of different types of cells, but we take a really sort of reductionist approach and we pretend there's only one cell, which is called the neuron. And in your brain, there's about 100 billion neurons, and they're connected in incredibly complicated ways. And what I'm showing you on this picture is a picture actually from my lab, and you're looking at a single neuron. There's like the engine of the neuron, that's called the cell body. And so that's like my body, and then it extends processes out and very, very far. So if I actually was a neuron, my processes would reach to Penticton and to Vernon, just to give you a sense of scale. Okay, so these are cells that have really long extensions, and they do that because those are really extensions on which communication occurs. So this is what Daryl was just talking about, that neurons communicate, and they communicate through a structure we call a synapse. And each of those little dots represent a synapse on that one neuron. So if you were to count those on even that one neuron, you can see there's probably a couple thousand in that single cell. Now in the brain, we have 100 billion neurons, and so if we have at least 1,000 of these things, you can see we actually have trillions of synapses in the brain. And these are changing constantly. They change a lot when you're growing up and developing, and they change also in adulthood. This is what the synapse looks like if we were to take one of those little dots and blow it up. And what it basically shows is there's one half, which is what we would call the pre-side of the synapse, and that's where all the juice comes from. This is the communication device that actually squirts stuff on to the other side of the synapse, chemicals that say it's time to be active. So that's what that's intended to show. So those green balls are the neurotransmitters that Daryl was talking about, and the other side of that cleft is a cell that's waiting to respond to those. And that's happening in your brain right now. As you're listening to me, there's little packets of chemicals going across saying, why is he wearing a black shirt, or whatever. You might think that this is a cute baby, and it is a cute baby, but it's also a human organism that's going under a huge amount of change. So the babies are born, they're not very good at very much, right? They don't play the piano, they can't write a poem, but they pretty quickly get better at doing a whole bunch of tasks. So what's going on in their brain? Well, what's going on in their brain is that when you're born, you have many, many more neurons than you actually need. A huge amount of neuronal death actually occurs early in development. And you also have many more connections than you need. And it's really important that you actually eliminate the connections you don't need. So when you're born, you have probably 3 times the neurons you actually need as an adult. So a process goes on to get rid of those. And we call that pruning, and we call getting rid of the synapse elimination. So there's a little movie, a cartoon of one of the leaders in the field actually, Jeff Lichtman, he's the guy on the left, showing you what really happens in the brain. You have this huge gemisch of connections going everywhere, and this huge pruning goes on, and so when it's all over, it says synapse elimination. So if you look in your brain on an MRI scan, that is what you will see. This is another picture from our laboratory to show you the neurons are actually amazingly good at doing this. This is over a course of a few hours of a living neuron in a dish, and we're just watching it, and we're saying, okay, we want you to undergo this pruning process, so we do some tricks that I can talk about later. But what you can see is there's this amazing clearing of these things. Boom, 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 boom. They just disappear. So it's a very active process. It's not passive withering that's going on in these cells. They're saying, I got to get rid of this, or there's going to be trouble. This is what happens in that baby we just saw the picture of. So that baby, let's say, was about 3 months old. And so in the newborn, you can see there's relatively few neurons. You get a little bit more, you get a lot more, and then by about 2 years, you're kind of at your max. And then with time, that pruning process I was talking about goes on. So you actually get fewer cells. So those big black dots are the cell bodies I showed before. And with time, you actually get pruning of the connections between them. And this shows going out to 6 years in a child. But it's not over. So brain development is still continuing. Does anyone know when brain development ends in humans? Pardon? Somebody? 25. 25 for boys, earlier for girls. Before I forget, what do you call a cow with no legs? Sorry. Thank you. Thank you very much. Okay. This pruning actually is a serious business. If you don't do it right, you have problems. So we now understand at least one of the major things that go wrong in conditions like autism is we have defects in pruning. There is data emerging that says there's a contribution at least to defects in pruning also in schizophrenia. So this is fundamental to how we wire a brain. And this process that I've described, it does occur in humans. It occurs in every species. From fruit fly to man, we're all really good at pruning neurons. And the reason is we have to learn which one of those circuits in the brain are important to us, and we're going to use those, and we're going to make them stronger, and we're going to get rid of everything we don't use. Okay? So this is a very, very important developmental principle in the way we develop. If we think about the adult brain, so now we're post-25, both boys and girls. The old thinking was that once you get to that age, it's all downhill. You know, your brain is made, nothing's changing, and it's really kind of a sad story. And it's partly true. We know with age there is cortical thinning that occurs, and so you know, kind of the common thought about the world's best physicists and composers are in their mid-20s to early 30s. There's probably something to that. Your maximal computation power as a human is probably in the age of 25 to 35, because then you begin a slow age-related decline. But that doesn't mean that good things aren't happening. While you're doing things, you're actually changing your brain. You're making new connections between different neurons in the brain. You're actually making new brain cells as a function of time. So for example, I don't know if you know this, but your nose, your ability to smell is determined by neurons in the brain. Those neurons in your brain are constantly turning over. You have to make new neurons all the time. Every day, you're making a few thousand neurons in the back of your brain, and they're migrating to the front of your brain where they become part of your nasal apparatus, and that's what makes you smell. And if that didn't happen, you wouldn't be smelling. So there's this kind of birth going on all the time. So our brains are constantly changing. And if we look at these two neurons in this picture here, and this is showing what I showed you before. This isn't out of my lab, it's another lab. But that's one neuron in red, another neuron in blue. They would be connected in a variety of complicated ways, and over time, they would change. In our brains, the reason things change is because we're experiencing new things. We're learning new things. And so this picture here of kind of the brain as a printed circuit board, you know, is a good analogy for what's happening, because the brain is composed of circuits. But what it doesn't tell us is the degree to which our brain is constantly adapting, always changing in response to new circumstances. You know, what happens to your brain when you read a book? Does it really change? Or when you learn to ski moguls? Or in my case, when you continue to kind of fumble your way through them? Or you have a really great golf game and you remember it 2 weeks later. How did that actually happen? Well, it's not magic. It's happening because there's chemical changes in your brain. And those chemical changes are represented in changes in those synapses, their ability to talk to one another, and the number of them, how many there are over time. This is kind of now seen as fundamental to neuroscience that, hey, guess what? You read a book, you change your brain. You watch a movie, you change your brain. You learn a joke, you change your brain. What do you call a cow with no legs? So I talked about pruning and synapse elimination in children, and I'm not going to have time to go through how this works in the adult, but it's a very similar mechanism. In neuroscience, when things are changing in the brain, we call it synaptic plasticity. And what that refers to is synapses are changing with time. And so it's a bit of a complicated diagram, but it's showing just what I showed you before, synapses that are changing over time. What this is showing is that there's a bunch of synapses on this one neuron, and this one are getting a signal that's saying, let's say, I've just read a book, okay? And then over time, what happens is these guys are going to get stronger, and over time, they're actually going to predominate. They're going to take over this whole area. So these synapses get bigger, and importantly, you have to get rid of synapses, even in your brain, in order to allow these to predominate. So there's names that I can talk about later about how this works and how we think this actually happens in real terms. But the bottom line is there's synaptic change occurring. And remember, you have trillions of synapses, and they're all changing all the time as you learn new experiences. So this is happening right now in your brain. And you say, so what? What does that have to do with Alzheimer's disease? Which is a completely legitimate question. The reason it's important is because the thinking in the field now is the first sign of Alzheimer's disease is synaptic failure. If you look at this diagram, this is actually from human patients in which there's been an examination of the synapse number in specific brain areas in people who appeared to be normal, people who had frank Alzheimer's disease, and people who have what we call mild cognitive impairment, which is often a precursor to full-blown Alzheimer's disease. And what you can see is there is a change between these three conditions. And if you look pathologically and ask what's happening, what you see is this synaptic loss really does correlate really well with impaired memory and seems to be a very early sign of Alzheimer's disease. Now, the problem is that synaptic loss is probably happening in a disease-causing way long before you're going to see any symptoms of Alzheimer's disease. Some of us in this room are probably experiencing slightly higher rates of synaptic loss right now that might be a precursor to Alzheimer's disease. That's not good, because if we look at what's really happening on the neuron, so this is the old friend I showed you a minute ago, and we take a look at this shaft here, normally what we would see is something that looks like this. We would see this shaft with these blobs coming off, and those are synapses. And what we're seeing in this diagram is one half of a synapse only. The way we've captured this image is you only see what's called the postsynaptic side. You don't see what's feeding into it. Okay, so you're only seeing half of it, but we do it that way because it makes it really easy to see what's going on. So you get a loss of those synapses, and so if my arms again are these neurites that are normally covered in synapses, those synapses bring communication, but they also help keep the neuron viable and happy and alive. And so when you begin to lose those synapses, what begins to happen is you get a retraction. So instead of being in Penticton and Vernon, I'm now in Lake Country and West Kelowna. And finally, I'm right here at the KGH. And because I don't have my supporting network of neurites bringing nutrients and important food really to the cell body, the neuron dies. The first thing is you lose your synapses. The second thing is you lose these processes, and that culminates finally in neuronal death. And that's a really important switch in the field. 20 years ago, we thought that Alzheimer's was a disease of neuronal death. Now we realize that neuronal death is a consequence of Alzheimer's. It's the end of the story, if you will. This is an Alzheimer's, two brains, one from a normal patient, the coronal section that Daryl was talking about, so this way. And what you can see is this very apparently healthy brain here. And on this side, this is a very advanced case of Alzheimer's, but it's obvious that there's been a huge amount of brain material loss. So in this case, you've lost the cell bodies, you've certainly lost many, many synapses, and you've lost the neurites. So what does this have to do with what I was talking to you about earlier? This person is not an early Alzheimer's sufferer. Her name is Beth Stevens. She's from Harvard, and she was really the first person to connect some dots that told us a little bit more about what's going on in the early stages of Alzheimer's disease. And that is, it looks like in the early stages of Alzheimer's disease, what's happening is an activation of those same pathways that occur during development. So during development, when we have all that pruning going on that I showed you earlier, it looks like in Alzheimer's disease, that process gets activated when it shouldn't. And over the course of 5, 10, 20 years, you don't need a lot of activation to cause some really serious problems. Beth was really the first one to show that this was something that was happening in the intact brain. She also showed in her first studies on this, by blocking some of those mechanisms that lead to that excessive pruning, you could actually lead to a retention of synapses that otherwise would have been lost, as well as increased function. And this is of course not in humans. This is in models of the disease, usually in her case in mice. That's kind of where we are now from a scientific point of view. We believe that Alzheimer's is a synaptic disease that then has long-term effects. And once the horse is out of the barn, once you lose those processes, once the neuron is gone, there's not a lot you can do about it. I mean, looking at that as a treatment option, for example, imagine that there'll be drugs that will lead to rebirth of those neurons. That's really a long shot. What we need to be thinking about now is how do you prevent that from happening in the first place? What are the conditions that will allow that to occur? And we got to get on top of this. Alzheimer's disease and Parkinson's disease, Daryl could show exactly the same slide from Parkinson's disease. These diseases are coming like a steam locomotive at us. In the next 20, 30, 40 years, we're going to be dealing with a huge increase in both of those types of neurodegenerative diseases. And it's because of the aging of the population as the primary reason. So there's a bolus of people who are going to come through the system who are going to be suffering with these diseases. And this is at a time while we're getting a lot better at treating other conditions: cancer, coronary heart disease, diabetes. At least the deaths associated with those diseases, the death rates are going down while Alzheimer's is increasing. For people who are interested in Alzheimer's disease, I really invite you to take a look at a website called AlzForum. It's a fabulous website for people like me who have pointy heads, people like you who actually have real jobs but want to know more about Alzheimer's disease. It walks through a whole bunch of information on Alzheimer's disease, from the basic stuff to the most complicated stuff. One of the interesting things they do, and this is a partial list, they publish all of the Alzheimer's disease trials that are going on, clinical trials that are going on. So right now, and this is a US-based site, but in the US, there's about 80 different trials that are registered right now for various treatments of Alzheimer's disease. There's a tremendous amount of effort around this, but most of these trials are actually focusing on patients that are showing signs of the disease or actually fairly along in the disease progression. And as I just told you, the problem is we got to get to patients before we know they're patients, which presents a really big challenge. So there's a lot of effort in going into understanding how we can diagnose Alzheimer's disease much earlier so we can intervene. But there's also trials going on, and there's one called the A4 trial, in particular out of Johns Hopkins University, in which they're enlisting patients that, from a variety of risk factors that I can talk about later, are at somewhat higher risk than the normal population. And they're treating these folks prophylactically. So you go in the trial, you have no signs of memory loss, you have no signs of mild cognitive impairment, and you can be enlisted. And these are folks between the ages of about 60 and 85. And they're going to follow these people for years and see what happens with the one particular treatment that they're using. So we'll see where that ends up. What about people sitting in the room? You're all healthy. I already told you you're attractive, which is good. But what can you do to reduce your own risk of Alzheimer's disease? Some of the same risk factors that Daryl talks about also apply to Alzheimer's disease, concussion being one that's emerged as being a really significant risk factor. But I want to talk a little bit more about building resistance. And you're going to hear a very consistent message. Exercise your body, exercise your brain, be socially active, de-stress, sleep, and eat properly, nourish your brain. My wife, who's sitting over here, Christine, teases me all the time about the fact that my first degree was in phys ed. It was actually in kinesiology. It's kind of advanced phys ed, really. And so I'm really interested in exercise. So I just wanted to tell you that exercise as a mitigating factor for various types of neurodegenerative disease, it really can't be overemphasized. The data on this is very, very strong, certainly from animal models, but now increasingly in human studies. Here, one study, this was done in rodents that had a model of Alzheimer's disease. I can just read out this. The evidence is clear that voluntary exercise, ... ultimately results in improved performance, including in models of Alzheimer's disease, Parkinson's disease, and Huntington's disease. It can elicit beneficial effects. UBC actually has a fairly big profile in this area, and there was something in the popular press October 26th of this year. Walking can reduce memory loss in seniors. This is real, and I really invite you to pay attention to it, and I'd be happy to talk about it more. So what can you do right now? You get moving. And this is a really, I think, a big community health issue, because we all know when you're 60, 65, getting moving might not be easy. You might have bad knees, you might have bad ankles, a bad neck. Things have gone wrong. So how do we as a society develop programs that allow you to get this benefits, but are recognizing the fact that we're not 20 years old anymore? So it's something we really need to be paying attention to. Use it or lose it, that refers to brain activity. It's absolutely true that if you are very active mentally, you're going to have a better outcome as far as one of your risk factors for Alzheimer's disease. Be social. Daryl also said this. Can't overstate this as well. One of the biggest risk factors for poor outcomes in men age 50 or more is the lack of a social network. So if you're in that age group and you're male, go get, keep some friends. Make sure you have a good group to hang out with. Being optimistic is actually not something that will mitigate the disease, but I just want you to be. There's a huge amount of work going on in this field. It's moving very, very quickly, actually. Daryl mentioned that we know a fair amount of Parkinson's disease because of the genetics. A lot of that has emerged actually in the last 15 years or so. A major event was the sequencing of the human genome, which I'm sure you've all heard about. We now have a total of about probably, 20 or 25 different mutations in separate genes in Parkinson's disease. And that's really important because it allows us to, basically in understanding how the cell works and how the machines in the cell work, are connected together. And so we can really build this map of that. We have some of that information in Alzheimer's disease. We're not as far along. And the only way we're going to get there is by doing cutting-edge research. My homework to you is to support basic research and make sure we can move the needle on that. So what that means is if everyone in this room wrote a letter to the Minister of Health and to our MP saying, we need to be supporting basic research in Parkinson's disease and Alzheimer's disease, it'll have a lot more impact than me saying it. You get to test me, but I really hope you do that. So the main lessons that I've tried to tell you, and it's completely legitimate, it's really true, the brain you have today is going to be different than the one you have tomorrow. Our brains are really adaptable machines. They're designed to change with circumstances and reflect our environment and allow us to respond to it. Millions of connections in your brain, millions are changing every day. So Alzheimer's disease, we now think, is initiated by just small changes in that activity, in the pruning mechanisms that occur between cells in the brain. What do you call a cow with no legs? Brain remodeling. So if you remember that little joke 2 days from now, or a week from now, or remember Daryl and I standing here, you can reflect back that your brain has changed. That's how that's happened. I'm going to stop there. Thank you.